That's much better. This is our presentation, short, medium and long-term studies of pigs fed GM feed ingredients. This was a collaborative project between Teagasc, which is the Irish Food and Agriculture Development Authority, and Waterford Institute of Technology. Our part in the GMSAFOOD consortium concerns testing the genetically modified BT corn in pigs on short, medium and long-term studies, and also we performed a small animal study with alpha-amylase inhibitor peas. I'll start by giving a brief outline of the presentation. I'll go through the objectives and rationale, I'll present the GM feed ingredients tested, and I'll move on to talk about results from pig feeding studies with BT Money. Then, on short term, that would mean 31 days, medium term, 110-day study, and a long-term transgenerational study. And I'll also present results from a pig feeding study with alpha-amylase inhibitor peas, which was conducted on short term, again, 31 days. I'll wrap it up with the overall conclusions, and we have a list of publications that came out of the project as a final slide. What were our objectives for this work package? Well, we wanted to evaluate the safety of short, medium and long-term exposure of pigs to BT maize and alpha-amylase inhibitor peas, and we wanted to identify any potential growth or health effects in response to GM maize and alpha-amylase inhibitor peas in pigs. By doing that, we also wanted to provide data for identification of potential biomarkers that can be used to predict any harmful effects of BT maize or alpha-amylase inhibitor peas following market authorization. Why did we do that? Well, because there are only a few studies conducted in pigs on investigating the effects of BT maize. Pig meat is one of the most widely consumed meats worldwide, and maize would be a major feed ingredient. What did we test? Well, the already famous BT Money 10 maize is expressing the cryo-1AB gene from Bacillus thuringiensis. This leads to production of the cryo-1AB protein, giving it insect-resistant to this guy, to the European core borer. At carbohydrate analysis, we observed a 2.5 percentage units lower enzyme-resistant starch in BT maize. We also performed proximate analysis, amino acid analysis of the maize limes involved, of the isogenic and BT maize, and we found that the differences are well within the published range, and by this I mean the OECD guidelines for new maize varieties, ILSI guidelines and FAO guidelines. To move on, alpha-amylase inhibitor peas express the AAI gene from the common bean, which gives them insect resistance to the pea weevil. The GM and isogenic pea seeds were provided by CISRO Australia, and before inclusion in the pig diets, we ground and heated them at 95 degrees for 15 minutes. For both BT Money 10 maize and AI peas, the GM and isogenic counterpart were grown over the side-by-side in Spain. Mycotoxin analysis revealed that mycotoxin levels were below the maximum allowable limits as set by the European legislation. No pesticide residues were detected, and the presence of the transgenic material was confirmed in both of them. I'll start off by presenting results from studies involving BT Money 10 maize in pig nutrition. The short-term pig study, for the short-term pig study, we used 32 male pigs, which were weaned at around 28 days of age, were given a 6-day adjustment period, following which they were randomly they were blocked by weight and ancestry and randomly assigned to one of two treatments, either isogenic or BT Money 10 maize in diet at 39% inclusion rate for 31 days. The medium-term pig study, the 110-day study, involved 72 male pigs, which were again within the 28 days of age, were given a 12-day adjustment period, blocked by weight and ancestry again, and randomly assigned to one of the treatments, either non-GM or GMAs in diet for 110 days. And we also wanted to investigate a potential age-specific effect. So we introduced 2 crossover treatments which changed the maize source following 30 days of consumption from either non-GM to GM or from GM to non-GM. For the long-term pig study, we used 24 gilts. They were blocked by weight and insemination date and randomly assigned to one of 2 treatments, either non-GM or GMAs in diet at 74 to 86% throughout gestation and lactation. Then their progeny, following weaning, they were again blocked by weight and ancestry and assigned to one of 2 treatments, either non-GM or GMAs in diet for 115 days. This created a split plot design with sow treatment being the main plot and progeny treatment being the subplot. I'll give an overview of the growth performance first. I'll go through slaughter characteristics, intestinal histology, serum biochemistry, and I'll end by presenting intestinal microbial populations. Then, so as I don't take all the glory, my colleague Maria Walsh will be presenting the immune response, and Eva Gellinger will present the fate of Bt toxin and Cry1AB gene in pigs. For the short-term pig study, in terms of growth performance, we saw an increase in average daily feed intake following 31 days of feeding. However, this was not associated with any differences in average daily gain feed conversion efficiency or weight at day 30. One thing I'd like to add is that due to a large amount of data that we produce from these studies, I'll be only presenting the significant differences, so at a significance level of P less than 0.05. When we analyzed the diets, we saw a similar dietary composition. The increase in average daily feed intake we observed in the short-term feeding study was not mirrored by the medium-term study, as we observed no effects in the first 30 days of feeding or from day 30 to 110 or overall. So these were parameters that we measured: daily feed intake, daily gain, feed conversion efficiency, and body weight. For the long-term study, in gilts, the only difference we observed was higher body weight at day 56 of gestation. However, this did not persist to day 110 and was not associated with any changes in back fat measurements. For the offspring, I'll present the treatment first. As I said, there was this is a split plot experiment. So we have treatment 1, non-GM gilt, non-GM progeny, treatment 2, non-GM gilt, GM progeny, treatment 3, GM gilt, non-GM progeny, and treatment 4, GM gilt, GM progeny. We saw a treatment effect, a gilt treatment effect here on average daily feed intake, with progeny of sows fed Bt maize having a higher feed intake, and this was also higher for Bt maize fed pigs as well. This increase in feed intake for progeny of gilts fed Bt maize led to a higher average daily gain and a higher body weight at day 115. There were no effects of sow or offspring treatment on water consumption throughout this study. I'll move on and talk about slaughter characteristics. For the short-term study, we observed no effects on organ weight. We weighed the heart, kidney, spleen, and liver, and a similar lack of response was observed on the medium-term study. Again, 110 days. Likewise, in the long-term study, we observed no differences in fat percentage at day 80. This was measured on live animals using dual energy X-ray absorptiometry. Furthermore, we saw no differences in carcass weight and dressing out percentage. For the long-term study in the offspring, we saw no effects on organ weights and body weight at birth, or on heart or kidney weight and back fat depth at slaughter. However, at slaughter, following 115 days of feeding, we saw lighter spleens in progeny of sows fed the BT maize. However, this was not present at birth, and an immune response would be translated by an increase rather than a decrease in spleen weight. Furthermore, we did not, the differences we observe now in spleen weight were not observed previously in the medium-term study and were not associated with any organ pathology. To move on, liver weights were lower in progeny on the GM-GM treatment compared to non-GM-GM and GM non-GM. However, this was not different between the GM-GM and the non-GM non-GM, which would be considered the control. Furthermore, carcass weights were higher for progeny of sows fed the BT maize, and dressing out percentage, which is a ratio of the carcass weight to final body weight, was higher in BT maize fed pigs. Again, no evidence of organ histopathology was observed in the present study, which questions the biological significance of the observed changes. We also went and measured intestinal histology, and you have a picture here of the intestinal villi crypts and the intestinal section of the intestinal structure. We measured villus height, so the absorptive area of the intestine. We measured crypt depth there as an indicator of the production of cells in the crypt. We also measured the number of goblet cells per villus, and this would be, goblet cells would be mucus producing, and a higher mucus production would be observed in an intestinal challenge. This would be associated with a reduction in villus height and the deepening of the intestinal crypts. We saw on the short and medium-term study, so again 31 and 110 days, we saw no effects on intestinal histological measurements. On the long-term study, however, in the duodenum, we saw an increased crypt depth in progeny of sows fed the genetically modified maize, and this led to a lower villus height per crypt depth ratio. However, this change was not associated with any signs of inflammation in the mesenteric lymph nodes following histopathological examination, and no differences were observed in the jejunum or ileum, so further down the small intestine. To conclude now on growth, slaughter characteristics and histology, for the short and medium term, we observed no consistent effects on growth and organ weights, no differences in intestinal morphology, and for the long term, we actually saw a growth benefit in offsprings of sows fed BT maize. The differences in intestinal morphology were not associated with a reduction in growth as one would expect, and the differences in spleen and liver weight were not observed previously, were not observed at birth from these offspring, and so they must have occurred during the post-weaning period. And we did not observe such changes previously and were not associated with any organ histopathology. To move on to serum biochemistry, what did we measure here? Well, we measured serum total protein as an indicator of protein turnover in the body. We measured serum urea and serum creatinine, which are indicators of kidney function and would be increased with kidney dysfunction. And we also investigated liver enzymes such as aspartate aminotransferase, alanine aminotransferase, alkaline phosphatase and gamma-glutamine-transferase, which again would increase greatly with liver dysfunction. However, on the short-term study, we did not see any effects on serum biochemistry parameters. For the medium-term study, I'll start off by presenting the graph. As you see here, First, the blue bar represents pigs that were fed non-GM diets for 110 days. The orange bar represents pigs that were fed a GM-based diet for 110 days. And then light blue and yellow represent the crossover treatment, which changed diets following 30 days of feeding, either from non-GM to GM, the light blue, or from GM to non-GM, the yellow. You can see the differences on top of the columns, and on the y-axis you can see the units of measure for the specific parameters. What can we see here? On day 30, urea was lower for the non-GM GM treatment compared to all others. However, as you can see from the graph, this was within the normal range for pigs. The dotted red lines represent the normal range for pigs. Furthermore, urea, this difference in urea did not persist to day 110 of the study, and no changes were observed in serum creatinine. On top of that, an increase in urea would be indicative of kidney dysfunction rather than a decrease. To move on, day 110 creatinine was higher for the crossover treatments compared to the GM and non-GM treatments. However, no differences were observed between the GM and non-GM. The serum creatinine, this rise in serum creatinine was not associated with a similar rise in serum urea, and was not associated with kidney pathology, which we examined at slaughter. So we did histopathological investigations. Again, it was well within the normal range for pigs. On day 110 as well, serum total protein was lower for the GM, non-GM treatment, but it was within the normal range. It wasn't different between the GM and non-GM treatments, and was not associated with any organ pathology. Again, this questions the biological significance of these findings. For the long-term study in gilts, we observed no effects on serum total protein, serum urea, alanine aminotransferase, aspartate aminotransferase, gamma-glutamine transferase, or alkaline phosphatase at any time point. This means day 28 of gestation, day 110 of gestation, and day 28 of lactation. However, on day 28 of lactation, serum creatinine was higher in the GMAs-fed gilts. Again, was not associated with a similar rise in serum urea, and was well within the normal range for pigs. For offspring at birth, no effects were observed on any serum biochemistry parameters. To move on, I'll start presenting the offspring that were fed the GM or non-GM treatment following weaning for 115 days, the multi-generational study, and as I mentioned before, it was a split plot, and you can see on the top half of the bars, this is the gilt treatment, so non-GM and GM, and on the bottom half you can see the offspring treatment. This gives rise to 4 treatments: non-GM sow, non-GM piglet, non-GM sow, GM piglet, GM sow, non-GM piglet, GM sow, GM piglet. On top of the bars, the X and Y's represent interactions. If there is a star at the top half of the bar, represents a gilt treatment effect, and the cross at the bottom represents an offspring treatment effect. To move on to the data, on day 30, alanine aminotransferase was lower for the non-GM treatment. However, it was not different between the GM and the non-GM, which would be considered the control. Again, it was within the range and did not persist to day 115. On day 30, likewise, creatinine was lower for the GM treatment compared to the non-GM, and non-GM. And this induced a lower serum creatinine for progeny of sows fed the BT maize. However, although they are below the normal range, you can see the variability in serum creatinine, the normal variability, and it did not persist to day 115 and was not associated with a similar or with an increase in urea. And again, this was a decrease, whereas an increase in creatinine would indicate kidney dysfunction. On day 115, serum aspartate aminotransferase, this again is an indicator of liver dysfunction, of liver function, was higher for the GM treatment compared to all others, and this meant that it was increased for progeny of sows-fed the BT maize. However, this was not associated with any changes in, there was no histopathology observed in the liver, and again, it was within the normal range. Was not associated with other enzymes a rise in other enzymes indicative of liver function. Overall, gamma-glutamyl transferase was lower for the GM treatment compared to non-GM, GM, and GM non-GM. However, this was not different from the controls, and the same story within the limits was not associated with any histopathology and was not associated with any histopathology in the liver. To conclude on serum biochemistry, on the short and medium term, we observed no differences between the GM and non-GM. For the long term, we observed an increase in creatinine at day 28 of lactation in gills-fed the BT maize, and in their offspring, we observed a decrease in serum creatinine on day 30. However, this did not persist and was not correlated with any changes in with a similar decrease in body weight. On the opposite, the body weight was higher. The values generally remained within normal range, and the differences did not build up into a pattern indicative of organ dysfunction. Furthermore, no organ histopathology was present. We moved on from we continued and we wanted to investigate intestinal microbiology, so we cultured indicator of pathogenic bacteria, which would be the Enterobacteriaceae in the intestine. We also cultured lactobacillus, which would be indicator of beneficial bacteria, and total anaerobes, which would be the total culturable bacteria in the intestine. And on the short and medium term study, we saw no differences in any of these bacterial populations. We took samples at the end of the short term study and every 30 days for the medium term study. We wanted to do something more exciting, and we performed high throughput 16S rRNA gene sequencing on cecal samples. For the short-term study, these were taken at day 31 of the study. We saw no differences in the relative abundance of bacterial phyla in the porcine cecum, and you have a graph here showing the major bacterial families within the porcine cecum, their relative abundance. So there was no difference, as you can see, between these major families. However, from the 39 families detected, we saw an increase in Enterococcaceae and Erysipelotricaceae in the intestines in the cecum of pigs fed the GMAs. However, these bacterial families are very low represented in the porcine cecum. You won't be able to see them on the graph. They'll be somewhere close to zero here, and they're not found in all the animals. As you can see here, for the Enterococcaceae, they were found in only they were detected in only one of the animals fed the isogenic maize diet, and in 5 fed the GM maize diet. Again, their role in the intestine is not fully established, so the biological significance of these minor, although statistically significant changes, but minor, is not yet established. At the genus level, from 54 genera detected in the porcine cecum, we only saw an increase in Bifidobacterium. This would be associated with health benefits in humans. However, in pigs, their role is not yet fully established. As you can see, again, they're found at with low relative abundance and with low frequency of detection in pigs. Likewise, Blautia was lower as a result of feeding genetically modified maize. Again, the role of Blautia in the intestine is not yet established, and if we look at the relative abundance here, it's not a major genus, and is not likely to have a major biological significance. On the medium-term study, again, we performed high-throughput 16S gene sequencing, and we took the cecal samples on day 110. We saw no effects on the 15 bacterial phyla that were detected, no differences in the 36 bacterial families. However, out of the 49 genera, there was finally a difference. You can see here the non-GM GMAs fed pigs had higher relative abundance of Holdemania compared to the GM maize fed pigs. However, this was not different between the non-GM and GM. Again, Holdemania are not, the role of Holdemania in the porcine intestine is not well established. To conclude on intestinal microbiota, we saw no differences in culture bacteria, either indicator of pathogenic, indicator of beneficial, or total culturable bacteria. Cecal 16S rRNA gene sequencing revealed no effect on major bacterial taxa within the porcine intestine. And the minor differences that we observed may have been due to a higher feed intake, which was observed in the short-term study. I was talking to you about a lower enzyme-resistant starch content in the BT maize. That may explain some of the differences, but again, the variability was within the normal range reported for maize varieties. Furthermore, no intestinal inflammation was observed or no health abnormalities. So we conclude that the BT toxin is well tolerated by intestinal microbiota. I think I've said enough, and I'll leave my colleague Maria Walsh to take care of the rest of the presentation. Thank you.
Thank you so much. So I'm going to continue now talking about the results from the MONIC 10 study with pigs, and I'm going to move on to the immune response. We evaluated immune response under 4 different headings. The first one was we had help from our colleague Ava Gellinger, who looked for the BT toxin specific antibody production in the plasma. We also looked at hematology, white blood cell counts, red blood cells, and parameters associated with red blood cells. We used flow cytometry to evaluate immune cell population phenotyping. And we also looked at cytokine production from isolated immune cells. And cytokines are just small proteins that are produced from a number of cells in the body, including immune cells, which affect the behavior of other cells. So first, looking at the cry-specific antibody production, we evaluated this in both the short and medium-term study. In the short-term study, we evaluated it on day 29, and in the medium-term study, we looked for it at day 30, 60, and 100. So I'm just presenting the short-term study data here. From the 2 graphs, you can see we looked at IgA production and IgG production, and this was in the non-GM maize-fed pigs, the GM maize-fed pigs, and also there was a control in there which was a non-maize-fed pig. And it's very obvious that there was no IgA or IgG production, and we found a similar result in the medium-term study. Moving on then to the hematology for the medium-term study, again we looked at differences on days 30, 60 and 100, and on day 100 was the only time that we saw any differences. Again, this graph is similar to what Stefan presented earlier on, with the blue bars being the non-GM for 110 days, the orange bars are the GM for 110 days, and then the crossover is the light blue and the yellow. So it's non-GM for 30 days, followed by GM for 80 days, and vice versa. So for the lymphocytes, we saw a difference between the final treatment, which is the non-GM for 30 days, and it was different from both the isogenic for 110 days and the GM for 110 days, but was not different to the other treatment, which was the 80 days of GMAs. What we did see, however, was the lymphocytes on this day, and only on this day, that they were above the normal range for pigs. For the red blood cells, we saw a difference between, again, the treatment of the GMAs for 30 days. It was different to those fed GMAs for 80 days or 110 days, but none of these treatments were different to our isogenic control. And again, these were within the normal range for pigs. And we found no effect of treatment on all the other hematological parameters that we measured, and there was about 15 of those in total. So we also looked at hematology in the sows for the long-term study during gestation and lactation. The only difference at this time we saw was for red blood cells. We found a decrease in red blood cells in those sows fed GMAs And again, this was within the normal range for sows. But what we did notice, even though the study was not designed to look at reproductive performance, we found a numerical increase in the number of piglets born alive for the GMAs fed pigs. And we hypothesised that potentially this may have influenced the red blood cell count, because as during gestation, the amount of the volume of blood in which metabolites are dissolved in increases, and this is proportional to the number of developing fetuses. So potentially this may explain why we saw a decrease in red blood cell concentration. However, we saw no effective treatment on the other 15 hematological parameters measured. We also looked at the hematology in offspring at birth. We found some differences in leukocytes and granulocytes. They were lower in the offspring from GMAs fed sows compared to the offspring from the non-GMAs fed sows. And we found no other effects at this time in hematological parameters. So moving on now to look at the immune cell phenotyping. So using flow cytometry, we looked at immune cell populations at intestinal level, and we found some differences in B cells and macrophages. These were decreased in the pigs fed the GMAs, and we saw an increase in the CD4 positive T cells in those fed GMAs. We did not find any effects of treatment on the immune cell populations in the splenocytes in these pigs. For the medium-term study, we found no effects of feeding GMAs for 110 days on the CD4 and CD8 positive T cells or the double positive T cells isolated from peripheral blood mononuclear cells. In the long-term study, in sows during gestation and lactation, there were some differences in monocyte percentages on day 28 of lactation. There was less monocytes in the sows fed the GMAs, and also we found some differences in the double positive T cells. Again, these were reduced in the sows fed the GMAs on day 28 or day 110 of gestation and day 28 of lactation. And there was no effects on B cells or CD4 positive or CD8 positive T cell populations in the blood. Moving on then to the cytokine production for the short-term study. So we isolated PBMCs, which were peripheral blood mononuclear cells, lymphocytes, and also lymphocytes from the intestine. And we cultured these overnight, and they were either stimulated with a mitogenic stimulator or they were not. And I'm presenting the mean values of these treatments. So we saw, we saw a difference in IF and gamma from the IF and gamma production from PBMCs was lower in those pigs fed the GMAs. And we hypothesized that by nature of the transgene insertion in the GMAs, that the GMAs is protected from insect damage. So potentially there's less of an endotoxin load in this maize, and this may account for why there was less of this inflammatory IF and gamma production when these pigs were fed the GMAs. We found some differences in the IL-6 and IL-4 production from the splenocytes. There was a slight increase in pigs fed GMAs. These were quite small increases, so we questioned a little bit how biologically significant this response was, considering we didn't see any cry-specific antibody production. And in the ileum, again, we saw an increase in the inflammatory IL-8 and also an increase in IL-4. However, we saw a decrease in inflammatory macrophages in the intestine, and again, we didn't see an antibody response to the BT toxin. And we found no effective feeding GMAs and other cytokines, which were measured through TNF-alpha and IL-12. So for the medium-term study, we found no effective GMAs for 1800 days on IL-4, 6, 8, and TNF-alpha production from both resting and mitogenically stimulated PBMCs. And similarly, in the long-term study in the gilts, when they were fed GMAs during gestation and lactation, we found no differences on the same profile of cytokine production. So to conclude on immune response, so we found some alterations in immune response. We proposed that the immune response is non-allergenic as there was no Cry1AB specific antibody production, and potentially the GMAs prevented an elevation in the inflammatory Th1 cytokine profile. So we were also interested in looking at the fate of the Bt toxin and the Cry1AB gene, and as I mentioned before, this was carried out by our colleague in Hungary, Dr. Eva Gellinger, and she's going to be presenting the results of her findings this afternoon. So then to just conclude on the effects of feeding Bt maize in pigs, we found no adverse effects on growth performance, no differences in body composition, no structural evidence of intestinal inflammation. The Bt toxin was well tolerated by intestinal microbiota. There was no evidence of organ pathology or functional change, and while we did find some alterations in immune response, these were non-allergenic. So now I'm going to move on to talking a little bit about the short-term feeding of the alpha amylase inhibitors, peas, to pigs. So this study was carried out similar to the short-term study we conducted with the Monet-10 maize. We had 36 mixed-sex pigs which were weaned at 28 days of age, and they were randomly assigned to one of three treatments. The first was a non-GM commercial pea, the second was a non-GM isogenic pea, and the third was the alpha amylase inhibitor pea. And these were included at 25% of the diet. There was a 6-day adjustment period post-weaning, and then they were penned individually. It was a completely randomized block design, lasted for 32 days. And the initial body weight of the pigs was 9.5 kilos, and all diets were formulated to be isogenic and isonitrogenous, and all peas were heated to 95 degrees for 15 minutes. So just looking at the growth performance, there was no differences in body weight at the beginning or the end of the study, and we also saw no differences between treatments in average daily feed intake, average daily gain, or feed conversion efficiency. And it's just worth noting that the pigs grew exceptionally well during this period. We also looked at organ weights. These organ weights were related to body weight on day 32, and we found no differences in the weight of the heart, kidney, liver, or spleen between any of the treatments. We also evaluated hematology, and we saw some differences in hemoglobin and hematocrit. And while we did see differences, the values still remained within the normal range for pigs. And it's interesting because we saw a difference between the isogenic and the GMP, but we also saw differences between the non-GM isogenic and the non-GM field P, which really brings home what was discussed a lot yesterday, where differences can rise because of variation differences or varietal differences. We also saw some differences in mean platelet volume. However, this time it was a difference between the GMP and the field P, whereas the isogenic P was not different to either of those two. And we found no effective treatment in any of the other hematological parameters measured. We also looked at organ histopathology, and we found no evidence of pathology in the liver or kidneys of the pigs fed any of the P treatments. And no differences were detected in the mesenteric lymph nodes between treatments. However, numerous eosinophils were observed in all of the treatments. So to conclude on our alpha amylase P studies in the pigs, we found no effect on growth performance, no changes in organ weight, and the changes in hematology were not due to the AIP, and there was no evidence of organ histopathology, and this analysis is still ongoing. So for overall conclusions, the differences in growth performance and slaughter characteristics, the effect was not consistent across studies. Intestinal and organ structure and function, there was no evidence of histopathology or dysfunction. Intestinal microbial populations, there was no effect on culturable bacteria or the major cecal bacterial taxa. There were some alterations in immune response. However, these were non-allergenic, and for the AIPs, there was no effect on growth, organ weight, histopathology, or hematology. This is just a list of some of the publications that we've had so far from the work that we've been doing. Some of these are published, some of them are in press, and we're still working on a number of manuscripts from this work. Just to acknowledge the Irish team that have been working on this project. You've met Stefan already, and most of you are familiar with Patter. There's also Professor Paul Ross, Dr. Mary Ray, and Dr. Gillian Gardner. Just like to acknowledge the funding for this project from the GMSAFOOD. project and also from the Teagasc Walsh Fellowship Fund. Thank you.
Thanks, Maria. Thanks, Stefan. This presentation is open for discussion. For a few minutes.
Thank you for this excellent presentations. I was just wondering, I have seen in one of your papers that the kidney was enlarged somehow. And you were just looking for those kinds of effects on long-term process. So, but I think you didn't mention about that, or was that right?
So that was in the short-term study. We found it wasn't actually a significant effect. We found a trend towards an increase in kidney weight in the short-term study. However, in all of our subsequent studies, we found no difference in kidney weights, and that included another short-term study, also a medium-term study, and we saw no differences in newborns also, and the long-term study. So it's not really a consistent effect or a reproducible effect, and it wasn't statistically significant.
Thank you very much.
Any other questions? I have a question for Stefan. The 16S ribosomal RNA measurements.
Yeah.
What proportion of the microbial population do you think you measured in those high-throughput measurements?
Well, this is an area that is under constant development, and it's new data is appearing every month or every few months. I wouldn't know how many of the bacteria, but most bacteria would have been represented, or the bacteria that would have had a distinct function in the intestine would have been accounted for at that age. What we're probably not seeing with this analysis is, again, bacteria that are not clearly classified or for which the role is not fully established, and further research will try to tackle that. But as you saw, there was no effect in major phyla, families and genera. So anything that was, we detected microbial populations up to 0.01% of the bacterial load in the intestine.
Very interesting. Any other questions? Comments? Yes, another one? You're allowed another one.
Well, there is another study which was done last year, and they have found cytotoxicity on human cells of Cry1AB. Seralini group has done this. Probably you know about that. What do you think about that? I mean, you have completely different results with that group.
I'm not familiar with that particular paper. I might ask you to provide me with more information after this is over. Were they using in vitro models or? In vitro models. Well, we tested everything here in vivo. So we tried to have a comprehensive approach or a holistic approach. So we didn't only measure one parameter and stuck to that. We measured a wide range of parameters. So we saw no change, no presence of pathology of either liver or kidney pathology. The intestine was fine, there were no changes in architecture, so, and this was all in vivo, and the rate or the inclusion in the diet was between 39 and 86%, so as much as possible to make sure if we get something it's relevant. Yeah. This might be due to differences between in vitro and in vivo work as well.
Okay, so we can talk about that.
Yeah, definitely.
Well, I think we might finish on that note. We're on time. Thank you very much for being on time, and thank you both for a very nice presentation.